Okay, a minute ago we were talking about transcription in bacteria, and we mentioned the promoter, and then we mentioned the terminator, and of course the gene itself, the coding region, sits in between those two. And now we're ready to focus a little bit more on the actual promoter. Now, obviously, if the sigma factor is going to stick to the promoter, it has to recognize something in the promoter so that it knows, hey, that's where I'm supposed to land. Right there on that spot. That's where I'm supposed to go. Well, in the promoter, these areas are called consensus sequences. And a consensus sequence is just anywhere where there is a certain ordering of nucleotides that seems to be very common across species. So it's conserved, right? It's it's like a general consensus. What's the general consensus? That everybody likes science? Well, everybody likes science, right? So most people have these consensus sequences. These sequences or orderings that are the same, right? They're conserved. And in the promoters, there are several different ones. Now, the most common one is the TATA box, right? T-A-T-A, TATA, the TATA box. And the TATA box is generally about -30 base pairs upstream from the start site of transcription. From the very beginning of the edge of the gene, -30 base pairs upstream before the gene in the promoter, right? And but there are a few others. Now, the TATA box, it's like I say, T-A-T-A, but it's not always just T-A-T-A. It could be T-A-T-A with like an A or a T, and then maybe another A, and then another A and a T, whatever, but it's the TATA box, okay? Now, at about -35 base pairs upstream, there's sometimes something called a BRE element, okay? And it's made up of a bunch of G's and C's, like GC, GC, maybe a little GA thrown in for flavor, a little CG, CCG. You know, you get my drift. Maybe a GGGGGGCCCAGC, something like that, right? Now, also, right within the edge of the promoter, somewhere close to the start site, there might be an INR sequence, an INR, and that's made of C's and T's, okay? So CCC, CCCTT, or CTCTCTCTCTCT, whatever, right? But basically that's the point. And so these are the consensus sequences. These are the sequences that the sigma factor is going to look for as it crawls along the DNA, trying to find its happy place. This is its happy place, one of these places. And then the RNA polymerase is going to look for the sigma factor and stick to it right there. Okay, but let's go back to the terminator. Let's jump to the other side of the transcription unit. Okay, now in bacteria, this is usually a series of A's and T's, a long series of A's and T nucleotides in a row. And so it'll be AT, 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 and so on, right? And it'll be preceded often by this twofold symmetric sequence of DNA. So that way, that when the transcript is made, of course, it's not going to have any T's in it, so it'd be UA, 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 UA. You get my point, right? Anyway, it's going to form a hairpin loop, and that hairpin loop in the transcript made of these UAs, 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 UAs. Anyway, that hairpin loop is what allows the transcript to fall off of the DNA polymerase and be free. So if it can't fall off, then it's stuck, and everything just jams up. And then nothing's going to happen after that, okay? So the hairpin, what it does is it wedges open this flap on the RNA polymerase, and it helps it to relieve the transcript, okay? Now, I'm getting ahead of myself. I'm talking about flaps. I'm talking about parts. Let's go back. Let's look at the structure of the bacterial RNA polymerase. How does that work? Well, it's a protein, but this protein's pretty fancy. It has these movable jaws, right? And those jaws clamp onto the DNA. And when they clamp onto the DNA, they crawl along the DNA, right? And and they can pry the DNA apart, open it up, right? Now, of course, there's this sigma factor that we talked about, and it allows this, when it sticks to the RNA polymerase, it allows this flap at the back of the RNA polymerase to close. And when the flap closes, what that does is it forms an exit tunnel for the RNA to leave, right? So as the little ribonucleotides get sucked into the tunnel and hooked together in the right order, then they can leave through this flap in the back as one long stringy RNA transcript, right? And so as this RNA polymerase is crawling along the DNA, there's this rudder, okay? And that rudder is prying the DNA apart. And as it's prying it apart inside of the enzyme, if we could zoom in inside the enzyme, we would see this DNA-RNA hybrid inside. As our little baby RNA transcript keeps growing with the complementary base pairing to the DNA template. Okay? Now, before we move on, just want to make sure that everybody gets the whole idea of the promoter, right? These promoter sequences they're kind of asymmetric, and that's important because the gene is only on one side of the DNA. Now, within one little region of the DNA, it's double-stranded, right? The DNA is double-stranded. Which side is the gene on, right? I mean, we don't have a bunch of RNA polymerases hanging out saying, which side should I stick to? I don't know, right? Of course they know. And the reason they know is because the promoter is asymmetric. It's only on one of those two sides of the DNA, of the double-stranded DNA. So it knows exactly where it's supposed to stick, okay? But that doesn't mean the genes only exist on one side of the entire strand of DNA in a chromosome. No, maybe this gene is on one side, and then the next gene will be on the other side. It's flip-flopping back and forth. Really, that's the way it happens. It doesn't just always stay on one side, right? So it's going to vary from gene to gene. But the point is that the gene is only on one side of the DNA. Got it? Okay. Now, let's take a quick break. Then we're going to talk about transcription in eukaryotes, like you and me.
Buy it, use it, break it, fix it, trash it, change it, melt, upgrade it, charge it, point it, zoom it, press it, snap it, work it, quick erase it, write it, cut it, paste it, save it, load it, check it, quick rewrite it, plug it, play it, burn it, rip it, drag it, drop it, zip, unzip it, lock it, fill it, call it, find it, view it, code it, jump and lock it, surf it, scroll it, pose it, click it, cross it, crack it, switch, update it, name it, read it, tune it, print it, scan it, send it, fax, rename it, touch it, bring it, pay it, watch it, turn it, leave it, start, format it, buy it, use it, break it, fix it, trash it, change it, melt, upgrade it, charge it, point it, zoom it, press it, snap it, work it, quick erase it, write it, cut it, paste it, save it, load it, check it, quick rewrite it, plug it, play it, burn it, rip it, drag it, drop it, zip and zip it, lock it, fill it, call it, find it, view it, code it, jump and lock it, surf it, scroll it, pose it, click it, cross it, crack it, switch, update it, name it, read it, tune it, print it, scan it, send it, fax, rename it, touch it, bring it, pay it, watch it, turn it, leave it, stop, format it. Technologic, 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 technologic. Buy it, use